Hematopoiesis. So hematopoiesis, by definition, is the process involved in the formation, development, and specialization of all functional blood cells. These mature blood cells are then released from the bone marrow into circulation. Sites of development. So in the fetus, blood cell production occurs in multiple sites, including the yolk sac, liver, and the spleen. In the adult, it primarily just occurs in the bone marrow. There are three phases of the hematopoiesis. So we have first the mesoblastic phase. This is the earliest phase where the blood formation occurs in the yolk sac of the embryo. Then we have the hepatic phase, which is the blood cell production will shift mainly to the fetal liver and spleen. Then we have the medullary phase, which is the final phase where the bone marrow becomes the primary site of hematopoiesis and continuing into adulthood. For the mesoblastic phase of hematopoiesis, age of development, it begins at approximately day 19 of embryonic development. The site of hematopoiesis occurs in the mesodermal layer of the yolk sac. The origin of primitive erythroblasts, excuse me, those primitive erythroblasts will develop in the central cavity of the yolk sac. There are different types of hemoglobin present during this time. So we have the Gower 1, Gower 2, and Portland. And these are embryonic forms of the hemoglobin specific to just this phase. The hepatic phase of hematopoiesis, so the age of development occurs anywhere between 5 to 7 weeks during the fetal development. The types of cells present during this phase are erythroblasts, granulocytes, and monocytes. They begin to start colonizing multiple organs, including that fetal liver, which is the primary site, the thymus, the spleen, and the placenta, and eventually these cells will migrate to the bone marrow as it starts to develop. There are different types of hemoglobin present during this phase as well, and you have fetal hemoglobin or hemoglobin F and adult hemoglobin or hemoglobin A. The peak hematopoiesis will start to occur around the 3rd month of fetal development, and the organs involved are your spleen, kidneys, thymus, and lymph nodes. The medullary phase of hematopoiesis, so age of development, this occurs between the 4th and 5th month of fetal development. The site of hematopoiesis during this phase is it takes place in the medulla or the inner part of the bone cavity or the bone marrow. The primary site, so by 24 weeks, around 6 months gestation, the bone marrow actually starts to become the primary site of the hematopoiesis. There are, once again, hemoglobin F and hemoglobin A present during this time. This is just a chart that kind of shows the differentiating hemoglobin levels and where they are, where blood is being formed during that time. So adult hematopoietic tissue, so we have organs and tissues that are involved, and that's the bone marrow, lymph nodes, spleen, liver, and thymus. Blood cell development, so erythroid development is the formation of your red blood cells. Myeloid development is the formation of your granulocytes, monocytes, and platelets. Then you have your megakaryocytic development, and that's the formation of your platelets. And you also have lymphoid development, and that's the formation of lymphocytes, also known as your T cells and B cells. We have primary lymphoid tissue. This includes bone marrow and thymus. This is the site where the T and B cells are derived and start to mature. Then we have secondary lymphoid tissue. This includes the spleen, lymph nodes, and mucosa. That's associated lymphoid tissue, also known as MALT. This is the site where lymphoid cells respond to foreign antigens and mount immune responses. So the bone marrow, there are different types of cellular components. So we have the red marrow. So hematopoietically active marrow is the red bone marrow. It's composed mainly of developing blood cells and their progenitors, so they're steps before, so kind of their babies, I guess, responsible for ongoing blood cell production. The yellow marrow, on the other hand, is the hematopoietically inactive marrow, and it is primarily composed of adipocytes or fat cells. It contains undifferentiated mesenchymal cells and macrophages, and it can revert to red marrow in certain conditions, like if there's been trauma, like a severe blood loss or something of that nature. Other cells that are found in the bone marrow are your stromal cells, and those provide structural support and secrete growth factors. Endothelial cells, these line blood vessels within the marrow. Adipocytes, which are fat storage cells. Macrophages, which are phagocytic cells that are involved in the cleaning up of debris and immune response. And then osteoblasts, these are bone-forming cells that contribute to the marrow environment. Osteoclasts as well. These cells are responsible for bone resorption or breaking down bone tissue, and they help to remodel bone and maintain the marrow space. So we have reticular adventitial cells, and these are specialized stromal cells that form a network supporting hematopoietic cells within the marrow. So red marrow cell development. So erythrocytes, red blood cells, are responsible again for that oxygen transport. Megakaryocytes are large cells that actually produce platelets. Granulocytes are white blood cells, including neutrophils, eosinophils, and basophils, and these are involved in the immune defense. The marrow circulation, so we have the vascular system within the bone marrow, and that actually supplies nutrients, oxygen, and helps remove mature blood cells into the bloodstream. The hematopoietic microenvironment, so it's a complex environment within the marrow, and it includes, as mentioned earlier, stromal cells, extracellular matrix, and growth factors that support the proliferation and differentiation of blood cell precursors. The liver. So the role of the liver in hematopoiesis, it is there for the embryonic hematopoiesis part. So in fetal development, the liver serves as a major site of hematopoiesis, primarily from the 6th week to mid-gestation. The liver hematopoiesis declines as the bone marrow becomes the dominant hematopoietic organ. Postnatal role. So in adults, the liver is not the primary site for hematopoiesis. However, it can reactivate hematopoietic activity under certain conditions. So if there's extramedullary hematopoiesis or something going on, it can start to kick back up if needed. The structure of the liver for in purpose of hematopoiesis. So we have the hepatic sinusoids, and these act as a vascular space where blood cells and plasma can interact with hepatocytes and immune cells. It allows for filtration, immune surveillance, and clearance of aged or abnormal blood cells. Hepatocytes, they play a supportive metabolic and regulatory role. The process break down products from red blood cell turnover, so it breaks it down, bilirubin from heme. That's kind of its process. Important cells involved are the Kupffer cells. These are specialized macrophages located within the liver sinusoids. Their roles are the phagocytosis of old red blood cells, the clearance of pathogens, cellular debris, and immune complexes, and they also participate in iron recycling after the red blood cell is degraded. Epithelial cells are hepatocytes. They metabolize hemoglobin breakdown products. They synthesize plasma proteins like albumin or clotting factors, and they are involved in the iron hematostasis, so the production of hepcidin. Let's see, so some pathophysiologic changes that are related to the liver and hematopoiesis. We have porphyrias. These are a group of metabolic disorders that can affect the heme biosynthesis. Liver and bone marrow can be involved in the synthesis of heme precursors, so an accumulation of toxic intermediates can cause neurovascular and cutaneous symptoms. Hemolytic anemia, so increased breakdowns of red blood cells leads to an overloading of the Kupffer cells with heme and iron. That will lead to elevated bilirubin, which can cause jaundice or yellowing for patients. Hepatic compensation by processing excess bilirubin and iron. Then you have your extramedullary hematopoiesis. This occurs when your bone marrow fails or is insufficient. So the liver, along with your spleen, will actually reactivate hematopoietic functions. This is seen in certain conditions like myelofibrosis, severe anemias, and certain thalassemias. And then we have storage diseases as well, so that can be genetic or metabolic disorders that involve accumulation of unmetabolized substances. An example of that would be Gaucher disease and Niemann-Pick disease. It can lead to hepatomegaly and impaired hematologic function due to organ infiltration. So the spleen, its function is to act as a filter of circulating blood. It is removing old and damaged blood cells and foreign particles. It is located beneath the diaphragm, behind the fundus of the stomach, in the upper left quadrant of the abdomen. The structure of the spleen, it has white pulp, contains lymphoid tissue that's involved in the immune responses, and that is rich in lymphocytes. And it also has red pulp, which contains vascular sinuses and splenic cords, which are responsible for filtering blood and removing those defective cells. The marginal zone, so the interface between red and white pulp, is important for trapping blood-borne antigens and initiating that immune response. To remove the red blood cell, we have a thing called culling, and that is an old or damaged red blood cells are phagocytized by the splenic macrophages with degradation of cell organelles. There's also pitting, which is where splenic macrophages remove inclusions or damaged parts of the red blood cell membrane while the red blood cell can still remain in circulation. So it essentially takes a bite out of it where that part is that it doesn't like. So some pathophysiological changes, so splenomegaly, which is the enlargement of the spleen, it can become palpable and can indicate some various diseases. You also have splenectomy, which is the surgical removal of the spleen. Benefits, it can relieve symptoms from certain blood disorders, it can reduce excessive red blood cell destruction, or they can do it to treat traumatic injury as well. Autosplenectomy is the functional loss of the spleen due to disease like sickle cell anemia, where the spleen becomes fibrotic and essentially non-functional. Lymph nodes, their function is to initiate the specific immune response to foreign antigens, filter particulate matter, debris, and bacteria entering via the lymphatic vessels. They are located throughout the body along the lymphatic vessels. Their structural components include germinal centers, which are sites within lymph nodes where B lymphocytes proliferate and differentiate. Follicules, which are areas containing resting B cells, they can become reactive during immune responses. Pathophysiologic changes, so it can happen in endonitis. It's an infection or inflammation of your lymph nodes. It often will cause swelling and tenderness. And then we have malignant tumors. That includes lymphomas and metastases from other cancers that can actually invade the lymph nodes. The thymus. So its function is a primary site for T lymphocytes, maturation and differentiation. It is critical for the development of cell-mediated immunity. The thymus is located in the upper part of the anterior mediastinum, and that's roughly at the level of the great vessels of the heart. Cellular distribution, we have predominantly composed of T lymphocytes in various stages of development, and it contains epithelial cells, dendritic cells, and macrophages that support T cell maturation. Size changes with age, so largest and most active during childhood, and it undergoes involution, so it shrinks and is replaced by fat after puberty. Pathophysiological changes, so this can be involved in diseases like thymomas and congenital immunodeficiencies. Hematopoietic stem cells and theories of their origins. We have types of stem cells. We have committed stem cells, and these are already destined to develop into a specific blood lineage. Then we have the non-committed stem cells, and these are undifferentiated, and these actually have the potential to develop into multiple blood cell types. So monophyletic theory is we have all blood cells arise from a single common stem cell, and then there's polyphyletic theory, and that's where different blood cells originate from multiple different or distinct stem cells. The lineage-specific progenitor cells, so we have common lymphoid progenitor. This will give rise to your lymphoid cells, so your T cells, B cells, and natural killer or NK cells. You have common myeloid progenitors. This will actually give rise to your myeloid cells, so your erythrocytes, granulocytes, monocytes, and megakaryocytes. So differentiation mechanism, there is symmetric division. Stem cell can divide into two identical daughter stem cells, expanding the stem cell pool. There's also asymmetric division, where the stem cell divides into one stem cell and one progenitor cell destined for differentiation. Apoptosis, which is the programmed cell death that regulates cell numbers and eliminates damaged or unnecessary cells during hematopoiesis. Then you have multilineage priming model. This is where stem cells simultaneously express genes associated with multiple lineages before committing to one specific pathway. So regulation of differentiation is you have extrinsic regulation that's controlled by external signals like cytokines, growth factors, and the bone marrow microenvironment. And then you have intrinsic regulation, and this is controlled by internal factors, including gene expression, transcription factors, and epigenetic modifications within the cell. So some morphological features of the hematopoietic cells. So cell size, this is going to vary depending on the cell type and stage of differentiation. Generally, Immature cells are larger. Nuclear to cytoplasmic ratio. This is the proportion of nucleus size relative to the cytoplasm. So a high NC ratio indicates immature cells. It usually has like a large nucleus and very little cytoplasm. Or a low NC ratio, and this is seen in mature cells where they generally have a smaller nucleus and more cytoplasm. Nuclear changes include changes in shape, chromatin, texture, from fine to coarse, presence of nucleoli, and nuclear segmentation during that maturation. And then we have cytoplasmic changes. There's alterations in your color, granularity, and appearance of inclusion as cells mature. Cytoplasm often becomes more abundant and changes from a basophilic blue color to kind of like an eosinophilic pink color as the cells mature. Stem cell cycle kinetics, so the capacity of cellular production. The bone marrow is highly productive. It is capable of generating about 2.5 billion red blood cells per kilogram of body weight per day, 2.5 billion platelets per kilogram of body weight per day, and 1 billion granulocytes per kilogram of body weight per day. So it's kind of a powerhouse. The mitotic index is a measure of the proliferative activity of cells, indicating the percentage of cells undergoing mitosis in a given cell population, and it will reflect the rate of cell division and marrow activity. The earliest hematopoietic stem cells, so we have surface markers. Let's see, we've got CD34+, CD38. I'm not going to read all these to you, but just to let you know, there are different stem cells, and these are all kind of the progenitor stem cells with them. You do not need to memorize these by any means. So some characteristics of cytokines and growth factors. So we have a group of specific glycoproteins. These are known as hematopoietic growth factors or cytokines. They regulate the proliferation, differentiation, and maturation of hematopoietic precursor cells. For classification, so growth factors can be classified based on their target cells and functions, such as a colony stimulating factor, interleukins, erythropoietin, or thrombopoietin. Functions of these is to stimulate cell division and lineage-specific differentiation and enhance survival and functional activation of mature blood cells. It has a positive influence. It promotes hematopoiesis by stimulating growth and preventing apoptosis of progenitor cells. The negative influence is some cytokines can inhibit proliferation or induce apoptosis to maintain homeostasis and prevent overproduction. So colony stimulating factors, their characteristics, they exhibit high specificity for their target cells. They're active at very low concentrations to help regulate that hematopoiesis. For nomenclature, they are named according to the primary cell line they stimulate. So for example, the GCSF is granulocyte colony stimulating factor that targets the granulocytic line. Functions, it can include hematopoietic colony formation in the semi-solid culture media, so in vitro, while typically specific, many colony stimulating factors can influence multiple cell lineage even beyond their primary target. Interleukins, so some characteristics, they have multiple biologic activities. Interleukins have diverse effects on immune and hematopoietic cells, influencing growth, differentiation, and activation. Synergistic interactions often work together with other cytokines to enhance or even regulate cellular responses. Amplification systems, so part of complex signaling networks that amplify immune and hematopoietic responses. And they are effective at low concentrations, so they can act at very low concentrations, which can enable precise control of cell functions. Erythropoiesis, so the site of erythropoiesis, as we mentioned earlier, it occurs, primarily in the bone marrow. We have some progenitor cells. Again, you do not need to memorize these by any means, just FYI on those. I would know erythropoietin or EPO though. That is the source. It's produced mainly by renal peritubular interstitial cells and to a lesser extent by the liver. Its function is to recruit the colony forming unit, so that one right up above it, from the earlier BFU, which is the burst forming unit compartment. It prevents apoptosis of the erythroid progenitor cells, and it can stimulate hemoglobin synthesis for red blood cell maturation. Leukopoiesis usually occurs primarily in the bone marrow. There are two types. First one is myelopoiesis. This is the production of myeloid cells, including granulocytes like neutrophils, eosinophils, basophils. It also does monocytes and other related cells. And then we have lymphopoiesis. This is the production of lymphoid cells, including your T and B lymphocytes and your natural killer or NK cells. Then you have megakaryopoiesis. Let's see, we've got factors that promote it. Again, you don't need to memorize these by any means, but the source of thrombopoietin, I would kind of know this, is primarily produced in the liver. Function of thrombopoietin is it regulates the production and release of platelets from megakaryocytes in the bone marrow. So therapeutic applications of hematopoietic regulators. So we have growth factors. It's used to stimulate production and function of blood cells in various clinical settings. We have chemokines, which play a role in cell signaling and trafficking. So there's potential therapeutic targets in hematologic and immune disorders. Plexiphores, a drug used alone or with those GCSFs. Its function as a mobilization agent to try to optimize collection of donor stem cells for transplantation.